Welcome to the COVID-19 Directors Colloquium. My name is Pat Fitch. I'm the Associate Lab Director for Chemical, Earth, and Life Sciences. Dr. Tom Mason, our Lab Director, asked us to summarize some of the R&D work that's been going on principally since March of 2020, but some of it predates March as well. Back in March, we kicked off a special LDRD process underneath of the Complex Natural and Engineered Systems Capability Pillar, challenge number 3, for those of you who are familiar with the pillar. And we turned this process around pretty quickly. There were about $125,000 per project, and we had almost 20 projects that got funded. Most of these projects were designed to start and end in fiscal year 20, so they've finished now, and some of the summaries you'll hear today are from these projects. We also formed as a lab the Special Office for COVID-19 R&D, or SOC-19. We have a leadership team for that. We've laid this out in a way that parallels something called the National Virtual Biotechnology Laboratory, which I'll talk about in a second. And our leads for that process, or that team, are Tony Taylor for manufacturing, Elizabeth Hong-Geller for lab testing, Srinivas Iyer for medical countermeasures, Kirsten Taylor McCabe for modeling, Irene Qualters for computing, Eric Hadberg for artificial intelligence and machine learning, and Duncan McBranch for industry outreach. And this team has been working continuously and are very appreciative. We, the whole group, are very appreciative of all the support we've gotten from across the lab in doing these R&D projects. And in addition to the R&D project, a special call out to our colleagues in operations and our collaborators in different parts of the government and industry that have been just essential to helping us understand how we can best align our R&D with their needs. So this new thing was formed by the Department of Energy in March called the National Virtual Biotechnology Laboratory. It includes all 17 of the DOE labs. All 3 of the NNSA labs are part of that 17. And it was designed to provide a single door to the National Lab Complex so that industry, academic partners, government entities, anyone who thought that the DOE might be able to help them in an R&D way could access those capabilities, that talent, through this NVBL. Our funding at Los Alamos for the NVBL totals around $15 million. I've called it out by area here. These are the specific projects they were funded. The work was roughly allocated by topical area to the biological and environmental research part of DOE, the basic energy sciences, and the computing part. You can see the dollar amounts. Most of these projects have a small amount of resources still going forward. Some of them are wrapping up right now. For instance, the manufacturing related work is wrapping up over the next week or two, and you'll hear some of those summaries as we go forward. A key part of how we design the R&D internally as well as with the NVBL is to look at what would improve our understanding, so more of a long-term look, and how to quickly help respond. Now, these are still R&D funds, and so they're not used to augment operations, but we did have a very near-term focus for our LDRD as well as the NVBL. And I'd say the things that we really managed to strengthen and leverage in this process since March are modeling at multiple scales. You'll hear quite a bit of that today. The use of additive manufacturing, you're going to hear some of that today. Bioinformatics and biological science as well. And then some really interesting multidisciplinary integration, which we're going to showcase today. Unfortunately, we don't have time to go through all the projects. There's numerous projects that are not going to get captured here. There's going to be a collection of articles in the 1663 magazine coming up soon that hopefully will capture an even broader array of these topics for you to see. And as I mentioned a little bit earlier, a key part to influencing R&D was our special connections and contributions from our colleagues in operations. The epidemiological work we've done together with multiple federal agencies and the state of New Mexico. The lab testing, where we stood up a lab, a CLIA registered lab here for Los Alamos is used for our staff, has also added tremendous value, and you'll hear about that in a few minutes. And then providing subject matter experts to support various operational entities, both at the lab and around the country. And then finally, a strategic plan that we've been working on, principally global security, Nancy Jo Nicholas and myself, for over a year now. We've been tuning that up and looking at revisions that may be appropriate given what the pandemic response has taught us about what we were good at, what we weren't ready for, how we might improve. And that is going to be part of a laboratory agenda item 2.7. So with that, I want to hand over the podiums to our presenters. We're going to end with ventilator research and development by John Bernardin. Preceding him will be Elizabeth Hongeller talking about laboratory testing, and preceding her will be Kirsten Taylor McCabe talking about global disease modeling and the Forecasting Center. Thank you, and please enjoy the presentations.
Hello, I'm Kirsten Taylor McCabe. I'm from the Global Security Program Office, and I'm here today to provide an update to the COVID-19 modeling response from Los Alamos National Lab. Some key facts I want to start with for the COVID-19 pandemic is that the coronavirus disease, COVID-19, is caused by the SARS-CoV-2 virus. It's spreading from person to person, mostly through respiratory droplets, but also aerosols and fomites. This is why there's been recommendations to wear masks. Spread's more likely between people in close contact with one another. This is why we have social distancing in place. Symptoms may appear 2 to 14 days after exposure to the virus. This is why we have self-isolation and quarantining in place when you have potential contact with someone who has COVID-19. Now, the last number that I saw was in the US, the known number of cases was about doubling every 3 months. And within New Mexico, last I saw was about every 2 months. About 8 months ago, when I gave a talk about the work that we were starting to launch in modeling over the COVID-19 pandemic, we were about 500,000 cases in the United States. Currently, we're at 9.28 million within the United States. But since that time, we have worked tirelessly to try and answer key scientific questions that would help in this decision-making process. What I want to highlight here in the honeycomb structure is LANL's core team disciplines for our modeling effort. As you can see, they really run the gamut of different disciplines from economics to information science, computational biology, biochemistry, and they've all come together through different directorates, different divisions to answer key questions around epidemiological modeling, disease surveillance and pathogenesis, understanding the structure and biology and the evolution of the virus, and then integrating all that data together to help communicate risk. One of the first efforts we had was a DOE-funded CARES project. It's a joint effort between Oak Ridge, Argonne, Sandia, and Los Alamos. It's an effort around pandemic modeling. Some of the bullets of the work done by the program is shown on this slide. They recognized very early on that the effective reproduction number, the R effective, was actually higher than originally estimated. They really did some deep dive modeling early on in the epidemic in such places as New York, Seattle, Italy, Wuhan, to understand what was driving the force of infection, so that you could understand what assumptions you had to put into your models, and you could pull out the parameters needed for this outbreak. They then use that knowledge in many different scenarios, and I'm going to give you highlights of that in this talk, from ICU loading to school reopening, to the impacts of opening bars and restaurants. They really worked on an end-to-end product to help for situational awareness and to understand the outcomes of different mitigation effects. Now, the work that I'm going to present today builds upon programs that we've had in place for a number of years, sponsors such as DoD, DHS, CDC, as well as DOE internal funding that we receive for this COVID-19 response through LDR projects and other projects like that. So I'm going to show you the breadth of work that we've been doing at Los Alamos since the beginning of the pandemic. Now, early on, the decision makers needed situational awareness. They needed to know what options were available to them, to understand the confidence of assessments that we were making. We had to pull in data that was available and understand the quality of that data and where we can make assumptions and check out the model performance. Two areas I'm going to start with to highlight are modeling for forecasting, statistical approaches for forecasting out the spread of the disease, and then simulation of different scenarios. So using multiple approaches, I'm going to give you some actual results that we've come up with at Los Alamos. So let's begin with forecasting. As you can see in the bottom bullet, we have a publicly available COVID-19 website that goes through COVID-19 cases and deaths forecasts. This is forecasts that go up to 6 weeks ahead for all US states, as well as many countries. This is one of the first 3 CDC-cited models for forecasting deaths due to COVID-19. When you look at the bottom 2 slides, you can see a 7-day average of forecasting out within the state of New Mexico, as an example. Again, this is done for all the states within the United States, as well as a 6-week forecast out. This has been used by such areas as the New Mexico Department of Health, and we have had a lot of inquiries in from all kinds of epidemiologists from across the United States who have been using this tool for situational awareness. This tool blends together epidemiological models looking at susceptible, infectious, and recovered states. And then it builds in the flexibility of machine learning to be able to forecast out, based on the behavior and the epidemic curve to date, what that might look for the next 6 weeks. On the right-hand side, you're going to see that this was actually a tool that was used in influenza to begin with. Then we were able to convert it over to COVID-19 forecasting. On this slide, you'll see concurrent hospitalizations on the top, and then concurrent ICU bed availability on the bottom. What was really important about this is last week, we were able to forecast out that New Mexico might exceed ICU bed capacity by November 8th. This is really important information for situational awareness and for planning for the state of New Mexico, but also this has been used and cited by the CDC and can be used for other states. Another area that we've used this forecasting is for non-congregate shelter. So when a person has COVID-19 and they need to have a place to shelter and quarantine themselves away from their family and friends, they need a place to go. By looking at the health region by country, looking at the daily cases in a health region, it can be by regions within a state, it can be at the county level as well. You can understand in the next couple weeks how many cases you expect. In turn, you can look at that county and predict out how many beds you might need in that non-congregate shelter housing. So this is a great way to facilitate planning action. What we'd love to be able to use this for is as you have a decrease in cases within a state, that you would be able to use this to plan when you have to bring down and consolidate those sheltering, because you don't need them as much because cases are decreasing. On the right-hand side, what you can see is COVID-19 across the state of New Mexico, a 7-day window comparison. You see the state right now, as of last week, is all highlighted in these bright purples. This is something that we don't want to see. It is where most New Mexicans are living right now in a county that has accelerating growth rates and a high per capita case count. So there's no counties as of last week that are decelerating right now. So it's a very important understanding of where the counties are. Are they accelerating? Are they decelerating? What support might they need? Now, to understand the impacts of assumptions and interventions, I'm going to go through 2 different tools. One is EpiGrid. So EpiGrid is a medium-grained epidemiological model. It's linking together geography globally. You have an image on the right of Haiti and a cholera outbreak, and then on the left of New Mexico with bordering states of Arizona and Texas. This brings together geography, disease and epidemic progression, and mitigations. It's a really fast-running tool. You can run a lot of different scenarios with it. It's been utilized in over 200 scenarios so far, just for the DoD. and it's spanning over 12 different diseases. So what I'm showing here is you can see globally in Haiti, you can see a state in New Mexico, you can see the case counts in the bottom over time, and you can also go down and start looking at the county level data. So to improve disease emergence for situational awareness and planning, this tool has been able to incorporate mobility data. Mobility as an independent estimators of the force of infection. So what you see on the top is mobility data over time. This is for the state of New Mexico. You can see when you have a shelter in place, you have this drop in mobility. And then as mitigation and public health orders change, you have an increase in mobility again. So you can look at this as an estimator of force of infection. You can also look at it from a aspect of looking at compliance to public health orders if it's a shelter-in-place situation. The tool is also fantastic to be used in what-if planning, such as school reopenings, which is shown in the bottom two pictures. These models can be seen on the newmexicohealth.org site for modeling that we were asked to do at the beginning of the outbreak, and again, subsequent when different case counts were going on, we reran the models to understand the situation at that time. So understanding the options that you have in reopening a school, from hybrid to all online to no mitigations and going back to school, we were able to model those different scenarios all the way down to the county levels to understand each county has a different spread rate within that county, and how would that affect a reopening within that county. It's also really good for understanding the differences between different districts. So if we go back and look here, you can see New Mexico, and then you can see a graph that shows Bernalillo in it. You can look at the infection rise rate in cases and deaths per county and understand what are the difference. What is one county doing better than another? What is one where we can have lessons learned? So what are those differences between different counties? We also have another tool that's been looking at impacts of workplace reopenings. So this is EpiCast. It is a hybrid agent-based epidemiological model. It allows the analysis of contact networks, travel. Very detailed intervention strategies can be looked at within this model. It has an explicit model that brings in together geography, but also demographic, different age groups. You can look at the impact of mitigations on different age groups. You can look at household size. You can look at worker distributions and community contacts and mitigations. So one way that this tool has been used at Los Alamos is in understanding how do we restart Los Alamos. If you bring so many people on site, 20%, 40%, 60%, what might that spread look like when you have a case, depending on how many people are back on site? Also, because we have the data available to us from Los Alamos, how do we look at the intermingling of people as they move from one facility location to another facility location? And how might that impact the spread? So spatial differentiation as a function of demographics and public health orders was also looked at. So you can take counties and cluster them based on similar income, population size, age, and education. So you can look at the United States and you can do this clustering analysis and then correlate between the cases in those areas and the change in public health orders. So, such as if you increase bar and restaurants, people being able to go to bars and restaurants as public health orders are relaxed, what might be the impact in those areas? What kind of testing would you need to increase? How many contact tracers might you need to have in place to be able to handle the surge that you might see in COVID-19 related cases? So bringing this together, we also had a tool called Median that looks at critical infrastructures like hospitals, hospital beds, supply chains, And then also can model contact tracing, testing, and diagnostics, and really get at the uncertainty of what is driving a pandemic outcome and what are the impacts of those mitigation strategies. So on the top hand image, what you can see is the impact of contact tracing, percentages of contact tracing that you can do over time, as well as the false negatives for testing. And then what you see is contact tracing and testing is vital for being able to understand and control the spread of the disease. But there's also still an acceleration in cases if you're not isolating and quarantining. So this is a way to quantify those and understand the primary drivers behind an outbreak. So a lot of the outbreak, when we put these mitigation strategies into place, we need to understand and be able to measure human behavior. One way that we've been doing that is through mining non-traditional data. This is the LDRD project from Los Alamos that's been mining Google and Twitter data. They've been looking at how do you understand sentiment of people for such things as vaccinations. If a vaccine was to come out, how many people might take that vaccine? That's very important to understand when you're making your assumptions for modeling. For mask usage, if there's campaigns out there that is increasing people using and being favorable to wearing a mask, what are those campaigns? Where are you seeing that favorable information, and how can you inform your communication strategies to improve upon what you're seeing in the sentiment? Another area that Los Alamos has been working on is the impacts of COVID-19 to behavioral health. So when you have a national disaster, historical national disasters, what we've been able to see is there's significant mental impacts to behavioral health, including depression, anxiety, domestic violence, and substance abuse. These impacts you can see in the yellow line that goes up and down in the top-hand side occur in these waves. And understanding from past national disasters where these waves might happen, where these vulnerable people might be affected during the epidemic is really key in trying to help people having issues with COVID-19 and having behavioral health issues. We've been analyzing data to understand mental health and understand if there's socioeconomic factors. How can we put a vulnerability index number and estimate vulnerability of certain populations so that we can determine what needs need to go to those areas, and how can you target interventions and strategies to help. Another area that Los Alamos has been working on, investing LDRD funds as well, has been understanding the transmission of outbreaks within a geographic location. So as you see in this phylogenetic tree, on the right-hand side, you can look at SARS-CoV-2 mutations that are reflected within the tree, and hence you can understand the transmission links that are occurring within a geographic location. So with this methodology that was developed at Los Alamos, you can identify transmission links. So you can identify if there's an outbreak that's coming into a geographic location, such as coming from a bordering state into New Mexico. If you could use this to look at transmission from international into the United States. You could look at outbreaks to understand if there's multiple introductions that are driving the infection within your geographic location, rather than just a single point source. And so this tool can look at the mixing among cases and showing that introductions of the virus into the state, like in New Mexico, are happening through mix of internal transmissions within the state of New Mexico, as well as from bordering states. We've been using our high performance computing power to look at such things as SARS-CoV-2 and why is it more invasive than SARS-CoV-1. So early on, we were trying to understand what was the difference with SARS-CoV-2, what could we understand about the biology of SARS-CoV-2. And what you can see here in the right-hand side is the red SARS-CoV-2 and the green human ACE2. The receptor binding domain of the SARS-CoV-2 has evolved. It's stronger grip on the human ACE2 than SARS-CoV-1. What you see is on the bottom hand side, you have a F486. That loop in the SARS-CoV-2 really forms this sticky anchor and maintains the interaction with ACE2 that you didn't see with SARS-CoV-1. So this is really important to understand why it was more invasive and understand something about the biology of SARS-CoV-2. In the upper hand side, what you see is this trimer image. And this was modeling done very early on in the epidemic, in the pandemic at Los Alamos. It was structural modeling that allowed us to understand that SARS-CoV-2 was acting more like a SARS virus than a MERS virus. And this really helped us parameterize our models early on and put assumptions into place that helped us with the modeling from the beginning, starting in January and February of 2020. We've been also looking at the spike protein. So a lot of talk has occurred around the SARS-CoV-2 spike protein. It's a protein that mediates the virus entry into a cell, and it's a prime target for COVID vaccines. So it's very important to understand how this spike protein might evolve over time. What we've been able to look at and see is in the spike protein, there was a single mutation, D614, that went to G614, and it has become the dominant form of the virus in a very small amount of time. So what you can see in this slide is by the midsummer of 2020, the G614 form became the globally dominant form of the virus in about a 3-month period. Now, there has been mutations that occurred in the past, and there will be mutations that occur in the future. So what we did is we built off our HIV database tools and developed a computational tool that can temporarily track mutations in the COVID-19 pandemic. You can see the link at the bottom of the slide, but this is really vital to understand and to keep track of. If another mutation comes, you need to understand this so that we can make sure that the vaccines being developed will be effective against the potentially form of the virus that is evolving over time. And lastly, what I want to talk about is some plume modeling that we've been doing. So we have a quick ChemBio plume model. It looks at the dispersion and deposition of particles. And what you see here in the top right-hand side is for a 100 micron droplet sneeze, how far would that travel? So we've been able to look at that quick plume model and understand how far that might travel in a specific scenario. Another thing that we've been looking at is outdoor dining, and is that safe? So when we had public health orders that closed down indoor dining, there was a lot of groups that looked at outdoor dining to be able to be part of the restart process. So what you can see is there's different ways that people might have space and be able to set up outdoor dining situations. In the upper right-hand side, you'll see an image where you can see this between two buildings, an outdoor dining site has been set up, and there's overhead screening structures set in place. So for some situation like this, deeper courtyards, you can use the model to understand that with weaker ventilation, you have airborne concentration of the virus similar to those found in an indoor eating establishment. So this helps for situational awareness. This can help people plan for the situations where they can do outdoor dining and do it more safely. So in conclusion, we do not know everything about the virus, about the pandemic, but we're leveraging our networks within Los Alamos with community tools, with our collaborations to help defeat this outbreak. Decision makers really need facts supported by science and data. We've been working on a layered approach of the different models to answer some of these scientific questions. We've been using multiple models. I have not been able to cover all the fantastic work done at Los Alamos, but I'm hoping I gave you a breadth of understanding of the areas that we've been working in. We need to maintain these capabilities for the next disease outbreak. And I really want to end on a thank you note. This shows the amount of work and amount of people that have been working on this modeling effort since the beginning of the pandemic. They have had such passion and they have made such scientific advances in the last 8 months, and I've been really honored to be able to work with them. Thank you.
My name is Elizabeth Hahn-Geller, and I'm the Deputy Division Leader of Bioscience Division. And today, I'll be speaking to you about COVID-19 diagnostic and next-generation lab testing. So as we all know, with the COVID-19 pandemic, it has posed a great threat to our public global health security as well as national security mission. So I wanted to give you some basic biology facts about SARS-CoV-2 before talking more about the details of our lab testing. So SARS-CoV-2 belongs to the family of coronaviruses that infect humans and animals and usually causes a respiratory distress. And already in the 21st century, we've seen 2 instances before this SARS-CoV-2 of coronaviruses crossing into human infection. And so that was the original SARS-CoV in 2002 and the MERS-CoV in 2012. If you look at the picture of a SARS-CoV virus shown here in the upper left, what you see is the virus structure surrounded by spike proteins. And it's these proteins which give coronavirus its name. So corona for crown, you see these dotted spike proteins surrounding the virus body. SARS-CoV-2 has a genome which is an RNA single strand. It's about 30 kilobases, one of the largest known RNA viruses. And the virus expresses about 29 proteins, a number of different functions that involve structure, replication, and host escape. And it's important to note that, like all viruses, SARS-CoV-2 isn't considered living. It requires the host in order to replicate itself and to infect. So what I'd like to talk to you about today is the different types of signatures that we look at in order to be able to detect the SARS-CoV-2, how we've established our diagnostic lab here at Los Alamos using the gold standard RT-PCR test, and then also talk about the different types of R&D that we've performed, existing tests that we can make more efficient, as well as to design better diagnostic tests. So we've set up the COVID-19 diagnostic testing laboratory at LANL back in March when it became apparent that COVID-19 was becoming a serious global health issue. And part of setting up this lab is that we had to get this lab CLIA registered. So this is a registration that's run through the CDC that states that we have the right processes in place with enough ability to handle human samples and then give actionable diagnosis of disease. So on the right side is a graph of all the testing that we've done at the diagnostic testing lab to date. So up until the middle of last week, we've performed over 7,000 tests and we've gotten 12 positives. What we've done so far is test approximately 350 to 400 samples per week. We're currently ramping up to be able to test 1,000 samples per week, and we're hoping to be able to do that by the end of this month. We're seeing all around New Mexico and the country that there is an increase in positive testing, and so we're going to try and increase our throughput here as well. So as I said, we've been using the gold standard nucleic acid amplification test protocol using RT-PCR. So RT-PCR stands for reverse transcription polymerase chain reaction, and it's a series of processes. So first, we would collect sample using a nasopharyngeal swab, and we do this at Auck Health. Next, the samples are brought back to HRL, our bioscience building, here on site for sample accessioning. So that's where we put the samples into our LIMS system, our database system, in order to be able to track what happens to the samples. Third is the virus extraction. So we take the sample and we break open the virus particles in order to extract the RNA. We turn that RNA into DNA, so that's the RT part, the reverse transcription part. And then we put the sample through the PCR, the polymerase chain reaction. So that's the fourth panel here. And this is a relatively involved process, so I'll just walk through it briefly. We separate the double-stranded DNA strands, and then we add a primer. So it's a short DNA sequence that will target specifically a gene in the SARS-CoV-2 genome, and that gives us the specificity of this assay. So the primer will bind to the DNA strands, and then we add an enzyme called a DNA polymerase. So that polymerase uses the primer as an initiation step in order to extend the DNA strand. The third component to this is the probe. So the probe also binds to the DNA further downstream from the primer, and the probe has attached to it a fluorescent dye and then also a quencher. When the dye and the quencher are close together, like in the probe, the quencher quenches the fluorescent signal. But one feature that the DNA polymerase has is that it'll cleave the probe when it gets to it, and that separates the quencher from the dye, releases the dye, and then we get a fluorescent signal. And that's how we are able to have this assay work, measure the intensity of the fluorescent signal. So then in the results, what we see is that when the fluorescence reaches above a background threshold, we get a particular signal. So for a sample with a high viral load, we're able to see this crossover above the baseline fluorescence at a lower cycle. So CT values is what we use to measure RT-PCR for this particular assay. So CT is a cycle threshold. So the more virus you have in your sample, the lower CT value you have. And usually if you have something like a CT value of 15 or 20, that's a sample with a lot of virus, whereas a low CT value, a low viral load has a high CT value of something like 35 to 40. So in the last couple of months, we've also been speaking on a weekly basis with the CDC and FDA. We're trying to provide reach-back capabilities for laboratory testing, various R&D type questions, in order for them to get independent verification to try and guide national standards. So on this slide, we're looking at two different things. So they were interested at the beginning of this pandemic when there was a supply chain issue. So all the supplies that are needed for lab testing, it was hard to get a hold of because all the labs were trying to purchase them at the same time. So they were interested in looking at different viral transport medias, these VTMs. So that's the liquid that's used to put the nasopharyngeal swab in order to stabilize the sample during transport to the laboratory. Another parameter they were interested in is storage temperatures. So at that time in the spring, summer was coming up, there was talk about trying to have folks self-swab at home. So if you were to take your clinical sample and leave it in a hot car for a couple of days, what would happen to the stability of the sample? So those were parameters that they were interested in. So we did this experiment in which we looked at 4 different viral transport medias. We made, there was a lab recipe we made here, a commercially available one, as well as buffers that were easy to get a hold of. And then we also looked at samples that were stored at 6 different temperatures. So everywhere from minus 80 to 37 degrees. And 37 degrees is the human host temperature, 98.6 degrees Fahrenheit. So we incubated these samples for 72 hours and then did our RT-PCR tests to see how stable the samples were. And so as you can see from the graph, so the x-axis is temperature and the y-axis is this CT value, this cycle threshold. We see that in all the different conditions that we tested with the different VTMs and the different temperatures, pretty much the samples remained stable around the same CT values between 33 and 35. And in this case, we were looking at our target genes were these two genes, N1 and N2, which are the nucleocapsid genes of the SARS-CoV-2. So this was interesting information. What surprised us was that even after 72 hours at 37 degrees, which is representative of a hot car, the samples didn't degrade or anything. So that was useful information for CDC and FDA to know. Another question they were interested in is this idea of pooling of clinical samples. So for example, if you have 100 people, you can do 100 individual samples to test. But if you have this sample pooling, you can divide the 100 samples into 10 samples of 10 each, and then do only 10 tests. And then if one of them turns out to be positive, then you can not have to do 90 of the samples and then test each of the 10 separately. So in effect, you're doing 20 tests to look at 100 people. So of course, sample pooling saves reagents and labor. It's great for low prevalence communities such as Los Alamos. After a certain point, if you start having a 5 to 10% prevalence, it might not be as useful of a tool. But if you have 1 or 2, then you can save a lot of labor. But for sample pooling, you also have a loss of sensitivity in testing and an increase in false negatives. And it takes a little longer to get to the final result. So you need to do 2 rounds of PCR in order to find all the positives. So the experiment that we set up to look at sample pooling that they were interested in is to examine the lowest viral load that can still be detected as a positive within a pooled sample. So here on the right on the table, we looked at different positive samples. So we have a pool A, a pool B, and a pool C. Each of them had different starting CT values. So for the pool A, the undiluted was about 28. And we made pools, basically 1 positive sample and 4 negatives for the 1 to 5 pool, or 1 positive and 29 negatives for the 1 to 30 pool, just to see when we start seeing loss of signal degradation. So in the pool A, even at the 1 to 30 pool, we still see reasonable CT values that look reproducible. But then as we go down with higher CT values, so that's lower viral load, in pool B, we started with a slightly higher CT, about 30. And then pool C, we started with 36, 35, 36. The red numbers, so all these experiments were done in duplicate. The red numbers means that 1 out of the 2 duplicates showed a degradation of signal. And then at the very bottom, pool C, where there are no numbers, we weren't able to get any results at all. So from these types of experiments, setting up different pool sizes and samples, what we were able to show is that we can still detect 1 to 5 pools with 1 positive if you have a starting CT of about 35. So you see that in pool C. But with a 35 CT, you won't be able to do a 1 to 30 pool. So more like a CT value of 30, well, you start to, can you still see a 1 in 30 pool where 1 sample is positive? So those types of experiments gave the CDC and FDA ideas about what the limits of this sample pooling could be. Another important aspect of trying to do efficient lab testing is to try and keep track of SARS-CoV-2 viral sequences over time. So like all viruses, SARS-CoV-2 is evolving as it goes from humans or to animals and to different populations. And so we've made efforts toward trying to standardize pathogen genome analysis during a pandemic. One of the tools that we have in hand is this EDGE bioinformatics platform. So EDGE stands for Empowering the Development of Genomic Expertise. It was a platform that was developed a couple of years ago to analyze all different sorts of genomics data. And it democratizes genomics with a user-friendly open source analysis platform. And it integrates hundreds of tools into easy workflows. So when the pandemic hit, we modified the EDGE platform in order to be able to analyze COVID-19 sequences as well. All over the world, scientists have been sequencing SARS-CoV-2 genomes that they've been able to get access to. So this starts with a diagnostic lab isolating the RNA and then putting the RNA genomes into a sequencing lab platform. There have been a large number of sequences available. So GISAID database currently has 174,000 genome submissions as of October. GISAID actually started back in 2008 as a repository for influenza sequences. And in addition to GISAID, we also have access to sequences through GenBank and through the SRA databases at NCBI. So with all of these sequences of SARS-CoV-2, we're able to do a lot of different types of analyses, some of which I've listed here. But the one I'll mention briefly is first one is to assess positive predictive value of currently available assays. So as I mentioned earlier, viruses also evolve over time, and that can pose trouble for developing assays that require specific primers and probes in order to bind to target SARS-CoV sequences. So part of the EDGE platform, what it can provide is this report card for PCR-based SARS-CoV-2 detection assays. So looking through all the different genomes of SARS-CoV-2 and how they evolve over time, we can look for different mutations and mismatches of the different primers and probes that are used by companies and universities that have been developed over the last couple of months. So if there are certain mismatches that start occurring as a virus evolves, then this tool will be able to note that, and then we can go to other assays or design different assays in order to have a better match to whatever virus is prevailing at a particular geographical location. So this is a bioinformatics search algorithm that provides this continual assessment so that we can ensure that SARS-CoV-2 doesn't evade detection with the assays that we're using. Another R&D effort that we're proceeding with is to try and design multiplex assays. So instead of just detecting SARS-CoV-2, we can try and use other primers and probes at the same time in order to also detect flu or RSV or some other viral assay at the same time as doing the SARS-CoV-2 assay. So there are a number of different considerations for trying to develop this multiplex assay. So the current limitation is that there's a high background fluorescence for the PCR assays that we use. So we need to somehow improve the dynamic range of these assays. And we're seeking to do this by combining properties of what's called these molecular beacons and this PCR assay that we use, also called the TACMAN assay. So molecular beacons have a lower background fluorescence. They're this stem loop structure that you see here. If we were to add this molecular beacon, this stem loop structure with a DNA sequence, what you see here on the lower left is that the ends where the stem is of the stem loop structure won't be able to hybridize with your DNA target. And so this is not compatible with the PCR assay. So what we're doing is to try and improve this assay is that we want to look for naturally occurring hairpin structures in the SARS-CoV-2 virus and other viruses so that when you do pair the molecular beacons with the target, we'll be able to get a complete match of the complementary strands so that we can use the TACMAN PCR assay. So this will require bioinformatics analysis and genome analysis of where these loop structures occur in the virus genome. And then other efforts we have going on, we have this fever pipeline, fever for fast evaluation of viral emerging risks. So this is a 3-part process where in the first step, the database, we collect SARS-CoV-2 sequences as we have been doing, and then we design new primers and probes that detect virus. In the United States in general, we've been focusing on the nucleocapsid N1 and N2 genes as our targets in SARS-CoV-2. So here, the fever assays are looking at a number of different targets in addition to the nucleocapsid gene. And then the third step is the test. So test these assays against spiked samples and patient samples. So to date, we've developed primers and probes for universal detection of all SARS-like viruses, and then also specific detection of SARS-CoV-2. As I mentioned earlier, the virus is evolving continually, and certain mutations will pop up over time. So we're also developing primers and probes that will recognize these mutations, also called SNPs, these single nucleotide polymorphisms in SARS-CoV-2. On the right here, it's just a test of the fever assays. So we show that we've developed primers and probes against the envelope gene on the top, and then this ORF1AB gene on the bottom, and that we're able to detect SARS-CoV-2 in spiked saliva at 1 copy per microliter, which is the same order as what our RT-PCR assays that we've been using in our testing lab has been. So to date, we've been mostly focused on looking at assays for detection in the RNA genome. So this has proved to be the gold standard. It's the most reliable and gives the least number of false positives and false negatives. There are other assays out there, and the one that has gotten the most attention lately is this protein antigen assay. So it's this fast assay. It takes 15 minutes. The RT-PCR assay requires specialized equipment as well as the staff in order to be able to handle human samples in a high complexity CLIA lab. The protein antigen assay is like a pregnancy test. It's a lateral flow assay, and it can be used by people without any specialized training. And the difference between the assays in the RT-PCR assay, we're looking at the genes in the viral genome. For the protein antigen assay, we're looking for the presence of particular proteins on the virus rather than the RNA. An important component in order to be able to make these protein antigen assays work is that you need specific antibodies that will recognize the proteins of the SARS-CoV-2. These antibodies can be used for different purposes. So on the right here, top, the proteins, the antibodies can be used for diagnostics. So if you have an antibody that recognizes a viral protein, you can use that along with some sort of detection system in order to say, yes, you have the virus. On the bottom right, you can also develop antibodies that can be used as therapeutics. So any virus that infects the human will likely target a human protein as well. For SARS-CoV-2, it's this ACE2 protein on the human that the spike protein recognizes. So if you can develop an antibody that somehow blocks the interaction between the spike protein of the virus and the ACE2 protein of the human, then you would have developed a therapeutic and blocked infectivity of the virus. So at Los Alamos, we have an affinity reagent team that develops antibodies that can be made to be specific to a target. So our folks looked at the molecular biology of the spike protein, and they selected a domain of the spike protein called RBD for receptor binding domain to try and develop antibodies to. So this would serve both the purposes of diagnostics and therapeutics if it's a good enough antibody. The antibody making process is a long, involved one. So here at the bottom, I've just given a short summary. So we have a library of all different types of sequences that will bind to different targets. So with this single chain library, we select for RBD binders, so antibodies that will bind specifically to the RBD domain of the spike protein. We then take what we find and convert them into human IgG. So this is the scaffold of what a human antibody would look like. And then we find pairs. So out of the antibodies that we select, we find pairs that will recognize different parts of the RBD domain. And so this allows one antibody to capture the target and then one antibody to produce a signal so that we can see that it's positive. So the results so far is that we've identified 20 unique antibodies against SARS-CoV-2, and then out of those 24 seem to be compatible pairs. So then the next step is to transition these antibodies into a lateral flow type assay that will allow us to test to see whether it's comparable to other assays available for detection of SARS-CoV-2. So those are some of the efforts that we have going on in the lab for lab testing. So I have some final thoughts here. The virus is evolving over time. Our lab testing capabilities must be able to keep up with the virus as well. So there's a lot of basic biology looking at genome sequences, looking at evolution, phylogenetic trees, in order to be able to see how the virus is evolving so that we can develop the assays to be able to still detect it efficiently. Next is the need to maintain R&D capabilities for the next pandemic. There have been series of pandemics over time, not only SARS-CoV-2, but influenza and other viruses. I think the national labs have a role to play here. We have a lot of interdisciplinary capabilities that can be brought to bear, understanding the basic biology of host pathogen interactions, as well as more operational, developing these detection schemes in order to be able to benefit public health needs. And then the third here, medical countermeasures. So there is also a group of scientists here at the laboratory who are developing small molecules, who are trying to use computational tools to down select already existing drugs to try and repurpose them as potential medical countermeasures for SARS-CoV-2. So that effort is going on as well. And finally, here on the right, I just wanted to thank you, all of the scientists and staff who have worked on this project over the last couple of months. There's a lot of cross-disciplinary capabilities that are brought to bear here, epidemiology, diagnostics, genomic sciences, and it's been great to see how we as a community have come together in order to try and defeat SARS-CoV-2. So thank you very much.
Hello, my name is John Bernardin. I'm a mechanical engineer in Engineering Technologies and Design Division. I spent the last 2.5 decades here at Los Alamos National Lab working on nuclear weapons, global security, and energy exploration projects. And back in April of this year, Tony Taylor and Pat Fitch approached me. They said, John, can you assemble a team to help us study this thing called intrapulmonary percussive ventilation to help us in the investigation of treating COVID-19 patients. And when they first mentioned it, as I said, most of my career has been in these different areas. And so working on life sciences, it was a new challenge, but at the same point, I thought we had the right capabilities and the ingenuity and creativity to tackle this problem. And so I assembled a team, and you're going to see a lot of names on this view graph. And everything I'm talking about is in credit to the ingenuity, the creativity, the special I guess the special energy that these people brought forward in a way that it absolutely blew my mind. I never anticipated 6 months ago that we were going to achieve what we did. And so I'm going to give you a whirlwind tour, hopefully in 15 to 20 minutes, give you a tour of what this team accomplished in 6 months. So bear with me, let's move ahead and I'll introduce some of the topics here. By the way, this was funded by the DOE Office of Science. So we're a great appreciation for the support that they've provided. All right, so before I get started on intrapulmonary percussive ventilation, and that's the last time you're going to hear me say that, I'm going to call it IPV for short. Too many consonants in there to keep repeating. First, let's talk a little bit about the lung. The human lung is a complex series of flow passages. There are, starting with your windpipe or your trachea, there are 24 generations of passages within the lungs. And you can see in the picture there on the slide, as those passages bifurcate and multiply, they get smaller and smaller. And that's depicted in the chart next to the picture of the lung. It works its way all the way down to these very small sacs called the alveoli. And it's there in the alveoli where all the magic happens in your lungs. That's where the oxygen and CO2 get exchanged. The oxygen comes into your blood system, the CO2 leaves. And that process is very important. All of you are experiencing that right now as you breathe and you're watching this presentation. Well, during the normal operation of a human lung, we're constantly producing mucus. It's generated in our lungs, and it's to remove impurities such as dust, small particulates. It could be invasive species that have tried to make their way into our lungs. And the lung functions to remove that mucus out of the passages. It actually migrates it upwards towards your esophagus, where you swallow it most of the time. Now, with what we found with COVID-19 patients, and in fact, anybody that has had a chest cold before, your lungs begin to fill up with mucus. As they're trying to not only get rid of the normal impurities, but now they're fighting an infection and trying to rid the lung passages of this invader. And so with COVID-19, there's a strong preponderance or a strong development of mucus that's generated, and the lungs begin to fill up. And as it does, the patients lose their ability to breathe and to do that oxygen and CO2 exchange. And so what the purpose of this project really was is to look at how can we improve the clearing of that mucus from the lungs to help patients perform that oxygen exchange with the CO2 and the blood flow. And so we looked on the commercial market. There are a number of different methods and techniques to remove mucus from the human lungs. These are generally home therapeutic devices. They're meant for short-term usage. If you have asthma, for example, you might use one of these to help. Maybe on a daily basis, you might use one of these for 5 or 10 minutes of treatment. And so here's just a few examples of commercial devices that allow that to happen. There's a chest wall oscillator. It's a vest that goes around your torso. It vibrates. It kind of frees up that mucus and allows you to cough it and expel it. There's what's called a flutter device. It's like a little child's kazoo. You put it in your mouth, you blow on it, and there's a fluttering ball inside of it that reflects acoustic signatures back into your lungs that cause that mucus to free up and allow you to cough it out. What I'm going to be focusing on today is this technique called IPV. It actually uses a pulsed aerosol that enters into your lungs, and through the deposition of medications, surfactants, and as well as the percussive effects, frees up that mucus. So let's take a little bit more of a deep dive into IPV. IPV normally is a short-term therapeutic home use or maybe a doctor's visit type approach to helping free up the mucus out of your lungs. Well, medical doctors had realized that, if you can combine this with normal ventilation, you might be able to, in the case of COVID-19, help patients in their recovery process by jettisoning or freeing up the mucus out of those clogged passages. On the right side there are a couple pictures to familiarize yourself with what this IPV looks like. In the center picture, there's a pistol-shaped device, and it starts in the bottom, and there's a nebulizer bowl. It's a gas jet atomizer, so it uses pressurized air passing over a film of saline and maybe some medications, creates an aerosol that is then jettisoned upward, gets caught in a venturi tube, and that is expelled out the end of the IPV unit and into the patient's mouth. And you can see on the right side there, there's a stream of cloudy aerosol propagating down the trachea and into the lungs of the patient. And then in the center picture on the bottom shows a ventilator flowing through a humidifier, passing then through a sequence of tubes where the IPV is hooked up, the flows merge, and the aerosol is carried with the normal ventilation into the human's lungs. And on the lower right, there's just an example of what this looks like from a sensor point of view. We have pressure versus time. The red line is the normal pressure rise and fall of a ventilated patient, and you see the rapid pulsing at a much higher frequency of the IPV percussive effect of the aerosol going into the lung passages. So what our team then attempted to do was use numerical and experimental approaches to begin to investigate the science and the engineering behind this and find out what parameters may make this function and work correctly, and to gain a better understanding from a very basic point of view of what's really going on and whether this is a viable technique that could be used in long-term treatment for a patient. And at the same time, we wanted to combine that with machine learning algorithms. It's a new science, a new engineering, where we're using these algorithms to speed up the ability to process information and to learn from data sets. And the hope is that in a medical treatment environment, the doctors can figure out the settings and the use of this technology in a matter of minutes rather than waiting hours or days to see if a patient's going to recover. We also brought to bear then, with that, various state-of-the-art tools that we have here at Los Alamos. Remember, I talked about this team, and I showed it on the first slide. They come from every walk of life in the laboratory. There's scientists, engineers, technicians. Most of them have never worked on a life sciences project before, but they all brought something to the table, and they pieced together what they thought were the tools that we needed to really characterize this. And when you really break this down, this is a fluid mechanics problem. There are a lot of attributes in here that we're very familiar with, just not in this particular application. So in addition to the modeling, we also did some experimentation. We used Pulmo, which is a miniaturized artificial human lung, to look at the alveoli response to this pulsed aerosol flow. We 3D printed flow channels of the human lung and instrumented them to look at how the aerosol would propagate, how the mucus would break up inside the lungs. And we even are setting up right now, as I give this talk, to use sheep lungs and to put dyes into the aerosol flow and then dissect the lungs to see where that aerosol propagates and where it deposits in the lungs, and then correlate that to how it might work in a human. We developed some novel optical techniques that I'll show you in the slides that come up, and developed some techniques to look at how the mucus can be upset and broken up in these little passages. So this slide is meant as kind of an orientation of all the things you're going to see in the upcoming slides here. This is quite an involved effort using a lot of different tools. And you remember that picture I showed you of the lung? There's 24 different generations, and that's what's shown in this slide here. As you look at the red line going from left to right, that attempts to segregate the lung into different sectors. Starting on the left, G0 is your trachea or your windpipe. G23 are your alveoli, where that gas exchange happens. And then in between are all these different passages of different sizes. And so we had to look at the tools that these engineers and scientists had available and said, well, which tools and which techniques can work in these different areas? We couldn't develop a model or do an experiment of the entire human lung. It's too complex. So we had to break it into pieces, and that's what this chart attempts to show, is that in the larger generations, we could use our standard CFD and acoustic modeling techniques. We could use targeted flow experiments to look at the aerosol flow and mucus breakup behavior. And then in the medium generations, the G8 through G21, we needed to look at the lung compliance and resistance, the fluid structure interaction. Your lungs are not static, they're expanding and contracting, and if there's mucus inside, their flow characteristics are changing. Every human lung is different, and so there are a lot of tiny passages, and those sizes and quantities vary between human beings. So we had to develop statistical models for these extremely small channels. And then I mentioned Palmo down at the smallest levels. And then shown on the bottom of that is an attempt to bring all of those models, all of those experiments together to form an end-to-end machine learning tool. The hope was to allow us to estimate what were the proper IPV and ventilator settings to help a patient safely recover from COVID-19. So the three questions that we posed to the team early on: can IPV enhance volumetric ventilation by aiding in the removal of mucus from the lungs? If so, what were the underlying physics? What was going on? How can we characterize this? How can we determine what's important, what's not? And then can we develop a useful tool that the medical community could efficiently use? If you're at all familiar with CFD or acoustic modeling, you'll know some of these runs that we use require high performance computing. They can run for days, weeks, even months. Well, if you're in an ICU on a ventilator, you've got a few days to maybe a week to get on the right track. And there isn't time to run all these simulations for all these patients. We've got to find a really efficient way to give medical personnel the information they need. Okay, so now I've introduced the project. I'm going to give you a quick overview of the tools that I've been talking about that we're going to bring together. In the upper right is that schematic that I showed, and the red box kind of gives you an orientation of where I'm at in that flow. So the first thing I'm going to briefly introduce is our high fidelity computational fluid dynamics model. This was done over in E1, and a number of different challenges in trying to model the flow passages in the lung. So we were fortunate enough to find a CT scan online, produced an STL file from that CT scan data. And from that, we were able to create the geometry that the CFD model required to basically, you're solving the Navier-Stokes equations, the equations of motion and mass and energy balance to look at the flow and the pressure characteristics within the lung. In this case, we were only looking at the first 9 generations, G0 through G8. That was all that we could manage in the time that we had for this modeling process. But we used large eddy simulations, looking at turbulence, looking at wall shear stresses, and how those all interacted in the breathing cycle. Again, this is a transient process, right? You're inhaling and exhaling. And here is just a snapshot of a few of the things that we're pulling out of this model. So we looked at different respiratory rates. And from that, we began to pull out wall shear stress. So in the lower left is a plot of shear stress during the breathing cycle. And in the lower right, we're identifying areas where we have peak wall shear stresses that might be damaging to the tissues within the human lung. And then the color contour plots, those are velocity contours. And the black arrows signify that there's rotational movement in those airflows within those passages. This is really important to understand, because when you inhale, the flow is not simply going in one direction into the along and backing out. It's actually swirling. It's a very complex flow pattern. And that's important to know. If you're trying to inject an aerosol deep into the lung, those aerosol droplets are evaporating, they're merging, they're depositing. And so you need to have some idea of how long is it going to take for these aerosol particles or droplets to transfer down into the deepest parts of the lungs. And this model gives us that ability to understand. Next, we needed to understand the viscoelasticity, or the compliance and the resistance of the lung to stretch and open up and then compress the airflow. And how does that change when a lung gets infected? How does that affect the pressure drop and the volume? So there's a lot of literature that's out there on this process. And we studied it and pulled everything we could from it and then expanded it. And in the lower part of this slide, there's an equation there, an analytical correlation that relates the pressure at the airway openings in the early part of your trachea, and it relates it to the volume of air that's going to come into your lungs, fill it up. The compliance is the inverse of resistance. So think of it as the ease of the lung in opening up as it's pressurized. And then V dot is the rate of volumetric filling of the lung. R sub M is the lung resistance, which increases when the passages are plugged with mucus. And then PMO is the PEEP, or the exhalation pressure. By understanding all those different parameters, we could build a library set of how the lung behaves from a fluid structure interaction point of view. And in the lower right is just an example of volume of the lung as a function of tracheal pressure during an inhalation and exhalation cycle. And that pattern will change depending on the condition of the lung. And that feeds into the machine learning model that I'll talk about at the end. We also needed to do some biological studies. As I mentioned earlier, we needed to understand, can we break up the mucus that's in these lung passages that are preventing patients from breathing? One thing I forgot to mention is that in the early days of this pandemic in the United States, you'll remember hearing about how the tragedy of what New York was facing. Their hospitals were overwhelmed, their morgues were filling up, people were dying left and right. If you ended up in an ICU on a ventilator and were incapacitated, the likelihood of surviving was something on the order of 20%. It was just dismal. And these people, what they were facing was basically suffocating. The mucus was filling up their lungs, plugging it up. You've heard about this from people that have died from pneumonia. It's a similar analogy to what was happening. So we really needed to be able to understand, could we break up this mucus? Can we get it out of the way to give these people a fighting chance? Can we improve that rate of recovery? And we were trying to harvest human mucus, but we couldn't produce it fast enough. So we had to, I give our scientists a lot of credit. They tried a number of different things to produce a simulated mucus, and what they found was that this carbopol 940, which it turns out to be the main ingredient in hair gel, and pardon the pun, they consider it the Goldilocks solution for our problem. It has very similar mechanical properties of human mucus. And a really important thing we discovered in this study was that the dynamic viscosity of this simulated mucus changed with frequency. We found that with mechanical disturbances, we could get this mucus to thin out and begin to flow. And that's extremely important if you're trying to clear it from a lung. And another piece of our puzzle here was to look at acoustics and turbulence modeling and experimentation to see, again, how could we remove this mucus that I talked about on the previous slide. In the center part of this slide was a pure experimental study where we looked at how acoustics could excite the lung passages. We're just looking at airflow going in and out of the lungs right now in this reference. And we found that IPV typically has an operational frequency of 1 to 10 hertz. It's actually fairly low. That frequency does not excite, does not resonate the lung passages. They tend to resonate at a much higher frequency, 3 orders of magnitude higher, and that's what's shown in the colorized plots there at 1.54 kilohertz and 4.77 kilohertz. We found that by changing the frequency of air pulsations, we could excite different areas of the lung or different regions. We're still investigating this, but we're hypothesizing that we may be able to combine acoustic signals with the IPV frequencies to target particular areas of the lung. In the lower part of this slide, now we get into the mucus breakup. So we were looking at how we could do shear thinning of the mucus using high-speed periodic airflow. And in that plot there in the center lower part of the screen, you can see that the mucus, the undisturbed layer, begins to get disturbed and shrink and be deformed by that that pulsed airflow. On the lower right is something even more interesting, and that was the Carbopol 940 test, where we coded a 3D printed simulated air passage. We put it on a small vibration table. Remember I said that the viscosity, the dynamic viscosity of the mucus would change with a vibration frequency. And so what we attempted to do was to vibrate the test section and then pulse it with IPV and see if we could disturb the mucus film. And in the lower right are two photographs. One is without any type of vibration ventilation. And the black layers you see there, that's the mucus film, and it's very stable. But notice what happens in the right side there. When we began to vibrate the mucus film and pulse it with the aerosol flow, we began to develop what's called a Kelvin-Helmholtz instability. It's like waves on an ocean. And as soon as you can do that, the mucus begins to move. And that's what your lungs need help with. They need to move it. Remember I said we need to move our mucus up into our esophagus where we swallow it and dispose of it. This indicates that these frequency techniques will work to break up and to move the mucus, which is so important to help these patients. We also did experimental studies to look at aerosol transport. And here are just a few examples of the things that we were up to over the past several months. We 3D printed and instrumented. In the center part of the slide is a simulated trachea that's instrumented with a number of different sensors. And in the center lower part of that slide is a more complex flow pattern. It's the trachea and then the first few bifurcations of the human lung. And again, these are all instrumented with pressure and humidity and temperature and sensors, and then optical sensors to look at the aerosol as it's propagating through. And then on the right side, we are in the midst of drying out and preparing some sheep's lungs to do some, as I said before, aerosol penetration with dyes, and then we'll dissect those to see how far the aerosol actually propagates in a more realistic lung environment. This slide shows the complexity of these tests. So you've got a ventilator, you've got an IPV unit, you got your instrumented test section, and a number of diagnostics. There's also in this picture a bellows, which represents the rest of the lung that couldn't be incorporated into the test section. And in the upper right is just a list of the different types of sensors we used. The most novel thing we created here was to really to look at the aerosol flow. Where was it progressing down into the lungs, and what variables were influencing it, either in a positive or a negative sense. And in the lower right there, we used a series of laser light sources and photodetectors to look at the light intensity. And in the oscilloscope plot, you can see there the yellow pulse is the very first optical sensor in a flow passage, and it picks up the aerosol as it's propagating in dry air. And then the next sensor picks it up. That's the light blue line, and you can see the pulse height is dropped. And finally, the purple line is indicating the third sensor response. And you can see this aerosol is changing. And in fact, what is happening is over a very short distance, the aerosol is evaporating. This was just an early test to see how we might use these optical sensors in a unique fashion to characterize this process. So here's just a snapshot of experimental data, things that we were able to find using these experimental tools. We found that humidity has a really, really strong effect on how this aerosol stays together and migrates down the lungs. Shown in that plot on the lower left is a family count distribution of quantity versus particle size for an aerosol. And what you can see there is that as we increased the humidity within the breathing cycle, the aerosol concentration coming out of the IPV and migrating down, in this case, a simulated trachea, would increase. Makes sense, right? You have more moisture, less propensity of the aerosol to evaporate and to disappear. In contrast, if you got dry air, like we have here in New Mexico, if you don't humidify that air, the aerosol will disappear and it'll never make it down to the deeper part of the lungs where it needs to go. Likewise, we found that increasing flow In both of those cases, if it was moist air, it would enhance the aerosol progression. If it was dry air, the increased flow would drop the ability for the aerosol to propagate. We also found that the IPV had to be synced with the ventilator. Remember I showed you that earlier pulse, the red line showing the inhalation and exhalation pressures, and then there was a higher frequency IPV pulse on top of that. In the plots on the right side there, the black line is volume versus time of the air going into the lungs in this one particular experiment. You can see in the first third of that cycle, that's your inhalation, and the latter 2/3 is the exhalation. What's shown below that is really significant. It's the optical sensor's signal, and the more negative the signal, the higher the aerosol concentration. And what you see is in that plot, there are a lot of big negative dips in the optical sensor signals that were placed along a bifurcated lung test section. And you can see real strong signals during inhalation, and during exhalation, there's hardly anything there. And it makes sense, right? You're pulsing an aerosol into a flow, and when it's being carried with the ventilator, it's making its way into the lungs. And when you're exhaling, you're fighting the backflow of the ventilator, and that aerosol is not going to make it into the lungs very efficiently. So what we discovered here is that it's very simple. You have to sync up the IPV with the ventilator. Okay, so those are all the different pieces, the different models, the experiments. They're all providing valuable information. But now what do you do with it? How can you compile all that information in a meaningful tool? And this is where the machine learning aspect comes in. This is linking everything, tying it all together. We had a very sharp postdoc on our team that figured this all out, and working with a few other people was able to piece together this fantastic algorithm that attempts to relate the expert, in this case it's a respiratory therapist, to a ventilator in the IPV unit, as well as a patient, and establish all of those relationships in that complete process of treating a COVID-19 patient. So normally in a hospital setting, a respiratory therapist sets the ventilation parameters based on the patient's conditions. They have to be careful because some ventilator conditions or parameters can actually damage the lungs. You can have too high a pressure, too high a flow rate, but the clinicians don't necessarily know what's going to be successful or not in a COVID-19 patient. It comes from experience and information sharing. As I indicated over the last few slides, we developed a lot of really neat tools to characterize how a lung behaves and how we might introduce IPV to clear mucus from these lungs. But we don't have time to do all these studies on a patient-by-patient basis. We have to have a fast-running tool that will spit out data and give a clinician real-time data to best set those settings. Remember, I was talking about a survival rate on the order of 20% back in the March-April timeframe in New York. We've got to, if we're going to use this technique, we've got to be able to give responders a chance to get on a path of recovery in a day or so. So we really need this tool, which has the capability to, once it learns all the relationships between these models and experiments, to run in a matter of minutes, maybe something on a cell phone, a Raspberry Pi board, something very fast and efficient. And so here's just an example of how this machine learning model works. It takes, for example, the computational fluid dynamics modeling output, and it takes all of that data and processes it with a statistical representation of the human lung. Remember, not every lung is the same. Some are infected at different levels. And so there's a statistical distribution of how that lung is going to behave as a fluid mechanics system. And so it takes all of the modeling, the probability distributions, takes that all into account, puts it through a neural network, and then spits out a predictive behavior of the relationships between parameters. And in this case, on this plot in the lower center of the screen, this is deep lung pressure as a function of ventilator pressure. And you can see that there's some trends there that the machine learning is beginning to deliver and discover. Where we're not yet at with this process is introducing clinical data to look at patient recovery. And that's really what you want, is patient recovery versus all the other parameter settings. And where do you hit that sweet spot of enhancing patient recovery? And that's where this, we hope that this machine learning will eventually take us. So I asked those 3 questions at the beginning to my team, and here's where we're at today. We're certainly not done. This has been a 6-month process, as I indicated. I think they've condensed work that could take a team of PhDs years to study, and they've condensed it into 6 months. And so we have some very positive preliminary findings. First, can we enhance volumetric ventilation by looking at the removal of mucus? Absolutely. We found ways to disturb the mucus to break it up. We see that in our modeling, that we might be able to do the same. The underlying physics, we've looked at turbulence and secondary flows, lung compliance, resistance. This is the first time that I've seen in all the literature that I reviewed and the experts across the nation that I've talked with. This is the first time that anybody's ever developed a comprehensive model of the entire lung system. And we're beginning to pin down what are the different physics, the different processes involved, and what are the influential variables involved in the breathing process. And then can we develop this machine learning algorithm? You just saw on the previous slide where the algorithm is kicking out predictive estimates on lung pressures. And so we're on the right track, but there's still a lot to be done. We certainly need more time to look at the mucus clearance and the aerosol propagation. We need more work on our submodels and our experimental results and find ways to incorporate that into the machine learning. And then we need to get clinical feedback data to bring this to reality, to put this in a medical operation where we can actually look at the usefulness of this tool. So there's a lot to be done, but I'm very positive, very optimistic that this team's on the right track, that I think everybody has felt that they wanted to fight back against COVID-19. And it's allowed this high-powered team to come together from all different disciplines and really spend a lot of extra hours when people are going home and having dinner or relaxing with their family. A lot of these people were working the extra hours. They were working weekends. I'm honored to have been their PI and to be involved in this effort. So I hope you enjoyed the talk, and I hope we lead to some positive responses to COVID-19 patient treatment. Thank you.